Hello, everyone. My name is Dimitrios Pantazis, and I am from Massachusetts Institute of Technology. I would like to thank the organizers for inviting me to this wonderful workshop. Today, I will talk about information decoding of MEG and EEG signals and how this can help us understand human cognition. First, let me quickly introduce the ideas behind decoding patterns in neuroimaging. Traditionally, neurovariate methods have been applied in neuroimaging data. These methods treat each variable, for example, a sensor or a source voxel, as an independent piece of data. However, multivariate methods have been gaining in popularity because they are more powerful. These methods extract information contained in distributed patterns of activity across multiple variables. Consider, for example, a single source or a single sensor. This source or sensor may not be able to discriminate patterns between two conditions, condition A and B here, when considered in isolation. However, when considering more sources and viewing the data in a multidimensional space, we can find a hyperplane that robustly discriminate the two conditions. This is exactly the idea behind decoding in MEG. Let me briefly review the procedures behind decoding of MEG signals. At every time instance, we concatenate the sensor measurements into pattern vectors of dimension m by 1, where m is the number of sensors. Let's assume that we have two conditions, A and B. Condition A may be responses to face images, whereas condition B responses to body parts. For each condition, we collect multiple trials by presenting the stimuli several times because of low signal-to-noise ratio. We can then train a classifier, such as a support vector machine, to discriminate between the two conditions using a cross-validation procedure. The classifier outputs a score, which is the classification accuracy. This can be 100% if the classification is perfect, or 50% if the classification is random, like flipping a coin. This procedure was done for a single time point. By repeating the same analysis across all time points, we obtain a decoding time course. This time course is very informative because it allows us to quantify the information encoded in the MEG measurements. These decoding tools are very popular both in MEG and the EEG community. For example, a recent work by Dubs in 2019 resolved the decoding time course of facial properties, gender, age, identity, and familiarity. This work showed that gender and age information emerged before identity information, suggesting a course to fine processing of face dimensions, thus providing new constraints on computational theories of face perception. In another work, Ziche and others revealed the dynamics of human object processing. For example, we showed that animate versus inanimate discrimination peaks at about 150 milliseconds in the brain from the onset of the stimuli. A work by Tang and others characterized the time courses of auditory source and space perception in the human brain using MEG data. And finally, work by Isaac and others showed how the brain can rapidly recognize objects despite transformations that alter their appearance. She presented stimuli that varied in position and size and showed that the brain constructs size invariant representations before position invariant representations. These were just some examples of decoding. These representations are really very useful, and we can go beyond category decoding by decoding, for example, individual stimulus images. This can also be useful because it gives us information about representational matrices. For example, in the work by Ziche and others, we presented images of 92 objects and then decoded every pair of images with each other. This procedure populated a 92 by 92 decoding matrix with each element representing the decoding accuracy between the corresponding pairs of images. For example, here, a hand versus a face image, as shown in the slide. Similar to the decoding time courses, these matrices are also dynamic, changing every millisecond. Let me show you a movie of these matrices. On the left, you will see how these matrices change over time. And on the right, you will see a plot of how these 92 images are mapped on a two-dimensional space using a technique called multidimensional scaling. This plot will show how images are clustered together by category. Initially, the matrices appear random because the stimuli have not been presented yet. At about 60 milliseconds, patterns begin to arise in the matrix, showing, for example, a clear distinction of face images versus other categories. On the right, you can see how images are grouped together by color, which indicates categorical membership of faces, bodies, and inanimate objects. The previous decoding matrices can tell us a lot about what representations are encoded in the human brain, but they can also be used for multimodal imaging, and in particular to couple MEG and fMRI data. Coupling MEG and fMRI responses is a difficult task because MEG signals represent electrophysiological activity, whereas fMRI signals represent hemodynamic activity. Thus, they are qualitatively very different. Combining the data can be achieved by neurovascular coupling models. However, this is a difficult task because these models are complex, and there is currently no consensus in the community for best models and their validation. Instead of using these complex neurovascular models, one can replace the MEG data with their representational matrices as computed before. Similarly, one can compute representational matrices with fMRI data. In this case, because fMRI has high spatial resolution, one can construct representational matrices in every voxel separately using a procedure called fMRI searchlight analysis. This is a very popular technique in the fMRI community, and for the interest of brevity, I will not explain this procedure here, but intuitively, the matrices are constructed by using the activation patterns of a neighborhood of voxels around each brain location separately. With this procedure, the data from both MEG and fMRI have the same form, the form of representational matrices. Thus, coupling the MEG and fMRI data becomes a straightforward task and can be accomplished by simply using Spearman's correlation. This results in a MEG fMRI representational similarity map, with its location indicating how similar are the fMRI data with the MEG data from a given time point t. Of course, one can repeat this procedure for every time point t, which will then produce a dynamic map of representational similarity. And these are the maps that I will show in the next slide. Initially, there is no significant representational similarity between MEG and fMRI. However, at around 60 milliseconds, you can see significant signals in early visual cortex. As time evolves, the signals move towards more anterior areas in the ventral visual pathway, activating areas such as the fusiform face area, which is responsible for the processing of faces, the lateral occipital complex, which is responsible for the processing of objects, and the parahippocampal place area, which is responsible for the processing of scenes. We saw how one can combine representations between MEG and fMRI data, but this analysis extends directly to other modalities as well. For example, we can couple representations between vision models, such as deep neural networks, and neuroimaging data, either MEG or fMRI. In the case of MEG, the representational matrices are a function of time. In the case of fMRI, they're a function of brain regions or voxels, as we saw before. In the case of deep neural networks, the representations can be computed as a function of model layers. Deep neural networks comprise multiple layers as they transform visual information from simple pixel-level representations to more complex categorical representations. In the work by Itzihe in 2016, we saw that the model representations share hierarchical structure with the human brain. Specifically, we correlated the MEG representations with the deep neural network representations, constructing MEG model similarity time courses. These time courses reveal that the first model layers correlated stronger with early MEG signals in that they had early peak latency. And the last model layers correlated stronger with the late MEG signals. Similarly, we correlated the fMRI representations with the deep neural network representations, constructing fMRI to model similarity maps. These maps revealed that the first model layers correlated stronger with fMRI signals in early visual areas, and the last model layers correlated stronger with fMRI signals in more anterior areas in the ventral visual pathway. Both MEG and fMRI results suggest clear hierarchical relationships between neural data and models. The previous work demonstrated correlational relationships between deep learning models and human vision, but this is just scratching the surface. In the last years, we have seen a renaissance in machine learning and computer vision. The performance of deep neural networks now rivals visual abilities from humans. Importantly, the relationships between brains and models are not just limited to correlations. Work by Striempf and others has shown that deep neural networks with better performance are also more similar to the primate brain in that they have a higher brain score, which is a benchmark based on brain data. This means that computer vision models are very valuable tools to provide insights into the computations actually performed by the human brain. And this is why I believe that bridging the gap between artificial models and MEG or EEG data is an important future direction. This will allow us to generate theories and explore the computations performed by the human brain. While we cannot be invasive with the human brain, we can be invasive in artificial models by manipulating their layers and computations. A recent work by Palazzo in 2020 exemplifies the benefits of linking brain data, specifically EEG data in this case, and deep learning models. They designed two models, an EEG encoder and an image encoder, that jointly map the corresponding data into vectors in a common embedding space. In this space, they forced the two vectors to be close when the EEG data were collected from the same image and far apart otherwise. By enforcing this common mapping, the EEG encoder learned more robust representations. As a result, the EEG classification performance had a boost from 48.1% to 60.4%. Therefore, such tools can have a significant impact in applications such as brain-computer interfaces. Of course, these brain deep learning models need large amounts of data to be trained, but I believe as a community we're heading towards the direction of collecting and analyzing large databases of neuroimaging data. Importantly, these models are trained. Once they're trained, one can manipulate their components to identify hidden relationships between EEG and model components. For example, the work by Palazzo showed saliency maps, which reveal how much information the neural data encode about the different parts of the image. In this particular image, the basket of a lady's head had the most impact in the EEG data. As we can see, the integration between EEG data and machine learning models goes beyond simple classification improvements. They can help us understand our data better and make connections with computational mechanisms of the human brain. I will conclude by a brief discussion on neurofeedback. With neurofeedback, we establish a closed loop where neural activity is measured online and is presented to the participant in real time through sensory feedback. In a recent work, we trained participants to enhance their attention to the left or the right visual hemifield. The participants successfully completed the task, and indeed, they had the desirable changes in their attention. We quantified this in a subsequent experiment where their brain MEG responses were stronger when viewing probe visual stimuli in the trained field. To conclude, I have described the importance of decoding brain representations from MEG data. While we have seen that MEG signals carry significant information, there is a critical need for non-invasive technologies that can capture even higher information content. I allude to optically pumped magnetometers, which can potentially be placed closer to the head, thus capturing MEG signals with a higher spatial resolution. Also, the research that links brain signals with deep learning models is growing rapidly. This will generate the need for large-scale data collection and analysis, affordable and portable technologies to record brain data in realistic environments, and so on and so forth. Last, neurofeedback and brain-computer interfaces create the need for effective methods to collect real-time brain data.